So now let's move to the second subtopic, which is disposable or single-use bioreactor technology. So what is this disposable or single-use bioreactor technology is all about? So the term disposable and single-use, they are used interchangeable. So sometimes people refer to disposable, sometimes people use single-use, but they refer to the same thing. Yeah. So by definition, disposable or single-use bioreactor is a bioreactor with a disposable bag or vessel designed for single use instead of multiple uses, such as the conventional stainless steel vessel or the glass vessel. And typically, this refers to a bioreactor in which the lining in contact with the cell culture will be plastic, and this lining is encased within a more permanent structure, typically either a rocker or a cuboid or cylindrical steel support. So if you have a look at the pictures on the right here, so these are some examples of disposable or single-use bioreactor. Yeah. So the first one here is made from plastics. Yeah, is a vessel, you can see. And the second one here is a disposable bag, which are normally used for animal cell cultures. And the bottom pictures here shows various sizes of single-use bioreactors, which are made from plastics. So basically, it's a concept whereby the bioreactors are made from single-use kind of material. I mean materials that are designed for single-use application, which is basically or more often than not are plastics. Yeah, so that is what disposable or single-use bioreactor is all about. So this technology has become an increasingly popular method of pharmaceutical and biologics production. So the reason why this technology is introduced is actually for mammalian cell cultures or animal cell cultures, because animal cell cultures are very prone to contamination. So the concept of disposable or single-use bioreactor is introduced so that the bioreactor used for the mammalian cell cultures can be free from contamination from previous batches. Yeah, because the vessels are completely new. So the way how the bioreactors, these disposable or single-use bioreactors come or reach the users, they are already sterilized and they are packed in plastics. Yeah, and they are to be opened in sterile environment. So once you have the disposable or the single-use bioreactor, so you have sterile media and you can put the media inside the disposable bioreactor and have your seed culture, and you can start your fermentation straight away. So it means that there is no autoclaving before and even after the fermentation. Okay. So, but even though the system or the technology has been introduced for mammalian cell cultures, we know that the mammalian cell culture are very different from microorganisms because they require longer incubation period, and the nature of one of the fishes is sensitive to contamination. Even though this technology has been introduced for mammalian cell cultures, but it also has been introduced to microorganisms or microbial cultures such as bacteria, yeast, and fungi, because the industrial player, they want to benefit from the ease of the disposable or single-use bioreactors because there's no cleaning and there's no risk of contamination. Okay, so based on that reason, the concept has also been introduced for microbial fermentation and not just for animal cell cultures. The recent technology of 3D printing has facilitated the customization of vessel and the agitator system to cater different needs of cultures. So these disposables or single-use concept, it does not exist in only one format or one size or one design. So various bioreactors manufactured by different designers or manufacturers, they have different sort of designs. So of course, based on that platforms or different platforms, the vessel should be different. Yeah, and the availability of the 3D printing has eased this by easily customize the vessels based on the needs of the platforms. Okay. Right, so you might be wondering why we want to introduce the use of single-use bioreactors. What are the advantages? So I've talked about the need for the animal cell cultures, because animal cell cultures are very prone to contamination, right? So apart from that, being like sterile, being sterile and being new, the disposable bioreactors, they do not require pre and post cleaning. So if let's say we are using conventional vessels like stainless steel or the glass vessels, what we have to do before the fermentation, we have to clean, we have to sterilize. And after the fermentation, also we have to clean and sterilize. So that is actually involve lots of steps, yeah, and it also means lots of money, lots of time and energy to be invested if you are using conventional bioreactors. So this can be reduced or eliminated by the use of single-use or disposable bioreactors. So that's one of the main advantages. And in terms of the cost saving, some estimates show that the cost saving of more than 60% with single-use system compared to fixed asset stainless steel bioreactors. So this is based on the research. So, and this will actually contribute to the overall cost reduction, yeah, for any production of any products. So that is one of the main advantages in terms of the reduction of the cost. And the second point is, as I mentioned just now, is the less risk of the contamination. Yeah, because especially for expensive products like therapeutic proteins, pharmaceutical products, they need stringent measures. They need much more strict measures compared to other non-critical products like fuel or enzymes, because those proteins or those pharmaceutical products, they are for human consumption. So stringent measures must be taken throughout the production process. So having said that, there is a need to invest on the use of vessels that can minimize the risk of contamination, such as disposable or single-use bioreactors. Right. Despite the advantages of the single-use or disposable bioreactors, there is a debate on the consequence of the use of the single-use bioreactors, because the use of these disposable things eventually will result in the disposal of plastics to the environment, right? So every time an experiment is done or a production is taking place, there is a waste of it, and there is a plastic waste. So this is one of the cons, yeah. But bear in mind that this technology is still evolving. It means that it is not yet finalized. It's not yet, it's not yet fixed. So there are rooms for further improvements. So perhaps the material for the bioreactor, for the single-use bioreactor, can be improved in the future by using biodegradable materials, yeah, whereby the materials at the end can be degraded. It's not just like accumulated as a permanent waste, yeah. So those are the things that can be considered in the future for the improvement of the disposable or single-use bioreactor. Right, talking about the environmental impacts of the single-use technology, although a complete life cycle assessment comparing single-use bioreactors and conventional bioreactors does not exist, but many ecological reasons are supporting the concept of single-use bioreactors. So comparing the two types, the single-use and also the conventional bioreactors, actually, although we see that on the surface, the single-use bioreactors may contribute to the problem of the plastic waste, but in terms of the energy saving, actually the single-use bioreactors may reduce the cost of the energy more than the conventional bioreactors. So based on what facts? The first point is based on the materials. The making of the conventional culture vessels from stainless steel or glass, yeah, in contrast to the plastics, actually it requires more energy. Yeah, so this is based on the survey or research that has been done. Yeah? Right. And then that is in terms of the material. In terms of the maintenance, yeah? So using conventional bioreactors, the culture vessel need to be cleaned and sterilized after each fermentation. So considering the cost of each operation, yeah, the cleaning needs what? It needs the use of autoclaving, right, for the sterilization purposes. And autoclaving, the sterilization process require the use of large amount of waters. The bigger the size of the reactor, the longer the autoclaving process will be, and the more water will be needed, right? So if you see the picture um, on the right side here, these are the pictures of 7.5 liter reactor. And these reactors need to be sterilized using large autoclave. This is a steam autoclave, which is used for autoclaving large equipments, such as large bioreactors. And the operation of this large autoclave is much longer than the normal autoclave, which you see in the lab, the one that looks like a washing machine, right? Because of the capacity is bigger, right? So having said that, it does not actually just consume more energy, but also water and also time, and also, yeah, the time, that is one of the critical factor. Yeah. So based on my experience, when I autoclave these two reactors, it took me like half a day for one cycle. So what I mean by one cycle is I have to autoclave before and after, right? So before the fermentation, I have to autoclave it for 4 to 5 or 6 hours, depending on the capacity of the things to be autoclaved inside. And after the fermentation, it takes another 5 to 6 hours for the sterilization to be completed. So on a working day basis, it took actually a day to complete the whole sterilization process. So it is actually a lot. And this is just 7.5 liter. So if let's say the reactor volume is increasing, like maybe 10 liter or maybe 20 liter, for those 20 liters or more than 10 liters, normally the sterilization will be in situ. So the steam requirement is actually quite a lot. The energy requirement is higher. Yeah. So it means cost, it means money. So that is actually to be counted when we want to compare the energy or the cost requirement for the conventional bioreactors and also the single-use bioreactors. Whereas the single-use bioreactors, you don't need to autoclave. You just throw the bioreactors after the fermentation. Of course, the waste should be handled properly, but there's no need for the sterilization process. Yeah. So it doesn't need water, it doesn't need any cleaning reagents, it doesn't need electricity to sterilize or to kill the equipment. I mean, like to kill the microbes on the equipment, right? So those are the things that need to be invested if we are using conventional bioreactors. So cleaning requires large amount of water in addition to acids, alkali, and detergents for cleaning. Yeah. And then the sterilization with steam at 121 degrees, 1 bar pressure requires large quantities of energy, large amount of water, distilled water. It's not normal water because the autoclave needs distilled water, right? So those are all the costs for running conventional bioreactors, which can be eliminated by the use of disposable or single-use bioreactors. So that's the comparison between the two types of bioreactor. So a comparison of the life cycle assessment of conventional and single-use bioreactor looks much more favorable for the single-use bioreactor as expected before. Yeah, and according to a report by Sinclair et al. In 2008, which was 12 years ago, the use of single-use bioreactors will help to save 30% of the electrical energy for operation, 62% of the energy input for the production of the system, 87% of the water, and 95% of detergents, all compared to conventional bioreactors. So this is the comparison, and it means a lot for the industry because everything means money, yeah, and also the time also means money, and as well as the manpower. So that is the motivation to support the use of disposable and or single-use bioreactor in fermentation.